So it gives me great pleasure to basically have the 2025 ABRF Award Lecture. So this award is given to scientists who have made a seminal contribution to biomedical imaging and also people who support that endeavor. So without any further ado, I'd like to introduce this year's awardee, Dr. David Deamer. So I'm going to read some of the specifics and then put one little anecdote in there. So currently, David is a research professor of biomolecular engineering at the University of California in Santa Cruz. He received his undergraduate degree from Duke, I'm not going to say the year, and his PhD in physiological chemistry from Ohio State. Now, Dave has always been interested in synthetic membranes. And everyone who knows Dave, even though he was not in my field, I certainly knew who Dave Deamer was. So we know that he was very interested in these synthetic membranes. Specifically, he thought and hypothesized that you could use it to sequence DNA by passing it through nanoscopic pores embedded in a lipid bilayer. So Dave, with his colleagues, demonstrated the feasibility of this in 1996. And then collaborative work, they proved a principle in 1999 by showing that the nanopores could distinguish between sequences of adenine and cysteine in RNA molecules. And this was where Oxford Nanopore was born. Now, what a lot of people don't know, and what I found incredibly surprising and really illuminating, was when the IPO for that company went out, it raised billions of dollars. And we're talking about a scientific instrument that was unheard of at that time to raise that amount of money from a IPO. And that was in 2005. So 2023 was a very good year also for Dave because he received 2 awards. One, which is not surprising, he was inducted into the Inventors Hall of Fame. The other one was he got the Golden Goose Award. And for those of you who don't know, the Golden Goose Award is given to somebody or a group of people that have done something that everyone believed was impossible. So this is a real sentiment to basically his dedication to this process and actually bringing it completely to fruition. So without any further ado, take it away.
Thank you kindly, Richard. This is a great conference. I walked in the door to the exhibits, and there was the company that you just mentioned, and I used their products. You'll hear about one of the products that we're using right now. This is Oxford Nanopore Technology, and me and my colleagues took a chance by spending 20 years of our lives working on something that might not work. And that our colleagues said, impossible. It just could never work, that you could pull a piece of DNA or RNA through a nanoscopic pore and get each base as it went by. But you're going to see it happening here, and you're going to see how we use it to answer one of the fundamental questions of life, and that is, where did nucleic acids come from, and how did they get involved in life on Earth, and maybe on Mars? You're going to hear a little bit about Mars toward the end of my talk. Well, the star of this show is up there on the screen. You haven't seen it yet, but I'm going to get rid of the text there. And there it is. A puddle? How can a puddle be a star of a scientific talk? I took that picture in Kamchatka, Russia. It was on the side of a volcano called Mount Matnovsky, and I'm going to tell you a little bit about that later. And the thing about that puddle is that it gets dry. It evaporates. The ocean doesn't get dry. The ocean's always there. But these little puddles go through cycles of wetting and drying. And there's a surprising property of wetting and drying as an energy source to drive polymerization. And that's what you're going to learn about from my talk today, is how we're using wet-dry cycles inspired by puddles to make nucleic acids from scratch, okay? So that's a nice take-home message. Somebody else, well known, better known than me, thought about this question 150 years ago. I'm going to let you read what Charles Darwin wrote in a letter to his friend, Joseph Hooker, in 1871. Darwin was very careful. He would not speculate in public, but he did take off his caution cap in letters to friends. So just 10 seconds, read what he said there, because it's quite significant. We're going to test that idea. We didn't mean to, but it turns out that we are testing that idea. We went to a volcano in Kamchatka to start the testing of it, and we're putting into a little hot spring, boiling hot spring water, basically what Darwin said there. There's going to add some phosphate. The ammonia comes in with amino acids that we add. There's light, heat, and electricity, although we're just using the heat of the wet and dry cycles. And we're not making proteins. Darwin did not know that proteins are polymers of amino acids, and he'd never heard of nucleic acids. You know, that just came along much later. But now we know how important nucleic acids are to life on the Earth. And the question is, when did they get started? How did nucleic acids come to be? Did life invent them? Maybe. Or did they pre-exist and were taken up by little compartments called protocells, and that was the first form of life? So you're going to get an answer to that, my best answer. You can believe it or not. It's up for grabs. It's not as though I'm telling you things we know for a fact. What you might want to keep in mind is something I do tell people. We can never know how life did start 4 billion years ago, but we can know how life can start, because we are stepping toward life in the laboratory, myself and maybe 100 other scientists around the world. And we're going to sooner or later know whether life could also have begun on Mars by a process similar to what I'm going to tell you about. So Darwin really was prescient in that, and we often use the idea of a warm little pond, but a warm little pond wouldn't work. Warm little ponds are not really good for research, because in Darwin's life, the ponds were surrounded by trees and grass, and there's living things in the pond. We needed to go to some place that was sterile, and the volcanoes of Kamchatka are sterile, as you will see. So the elements of life, I just want to introduce those and remind you, you probably know all these, but you might not have realized that they make up 99% of a living cell. So these are the cells that we use in our bodies, and they go all the way down to bacteria that have these 5 or 6 biogenic elements. And you're going to learn something very soon that is quite extraordinary about these elements. So it's a hydrogen, carbon, carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. Those are the 6 biogenic elements. Where did they come from? They came from interstellar dust that preceded our solar system. And stars and solar systems are formed from clouds of interstellar dust particles. And you're going to see one soon. In fact, there's one in the background there that you can see. These are the ashes of stars that have gone through their lifetime and exploded and given all of the atoms that they synthesized by nuclear fusion, nuclear fission, and those atoms now are strewn out into space and gathered together by gravitational force into what we call molecular clouds. Now that really is an interstellar dust particle. The U-2 aircraft would fly very high, and they would put out a sticky film and collect these particles and bring them back for us to see. Now the reason those particles are so important is that there is a layer of water. Water ice is on the surface, frozen on the surface of the particles in that molecular cloud. We know that for a fact because we can get a spectrum of water in the radio frequency range. Also in that ice is embedded simple molecules like ammonia, methanol, carbon monoxide, carbon dioxide. So these are the elements being brought to the Earth during the accretion of the solar system, of all the planets and our sun just starting to glow by nuclear fusion. So that's important to realize that that is the source of all the carbon, nitrogen, oxygen, phosphorus in this room, sitting there in the audience. You came from a thin film of ice on interstellar particles. So let's think a little bit different, a little bit longer about that. It's an amazing fact that 70% of the atoms in life and in our bodies are as old as the universe. The atoms of water, hydrogen oxide, the atoms of water are left over from the Big Bang. They didn't get caught up in star formation, and we have the privilege of using them in the water that circulates in our body. I'll bet you didn't know that you are 70% as old as the universe. Now, what about the other 30%? Well, those are the elements, the biogenic elements, that are made by stellar nucleosynthesis in stars. Part of the process that a star goes through during its lifetime is synthesizing these larger atoms like carbon and nitrogen and oxygen. So the take-home lesson is that dust, that interstellar dust that you see in that cloud surrounding a star that has died and strewn that into space, that is the source of life on Earth. And as we look up with Carl Sagan, we are stardust. So take that home, tell it to your kids if you have them, and this is something that still amazes me to think that we are part of the universe. We borrow a little bit of the universe to be alive here, sitting in this room. Okay, what do we need to make life? So we need three major things. These are the primary molecules of life. We need nucleic acids, and you see DNA double helix up there. We need amino acids. Those are the monomers of proteins. And we need amphiphilic compounds. Now that might be a new word for you. Amphiphilic compounds are in fact able to self-assemble into membrane compartments. So if you're going to have cellular life begin on the Earth 4 billion years ago, you'd better have a compartment for it to keep all of its systems, biochemical systems, in one place. If you don't have that, life could not begin. Now, I'll bet that everybody in this room has blown soap bubbles when you're kids. Maybe even now, I don't know. It's fun to blow soap bubbles and watch them float away. Soap bubbles are membranes made from an amphiphilic molecule called soap. And that's basically got a hydrophilic group on one end of the molecule and a hydrophobic group in the hydrocarbon chain. So that's why it's called amphiphilic. It loves two things. One end loves water, the other end loves to be in an organic solvent of some sort. So that's been part of my life throughout my career, is dealing with amphiphilic molecules and the fact that you can get them to self-assemble into membranes. And you'll see that again later in my talk. So here is Hawaii, except it's 4 billion years ago. Hawaii is a volcanic island. The Big Island has got two two volcanoes on it, Mauna Loa and Mauna Kea. And Kilauea is still active. It's spewing out lava right now as we speak. But that's what the Earth looked like 4 billion years ago before life began. We called it the prebiotic Earth. The atmosphere was nitrogen, a little bit of carbon dioxide, and no oxygen. And of course there's no oxygen. There's no photosynthesis. There's no plant life making oxygen for us. The oxygen that we're using right now in our metabolism was synthesized by cyanobacteria about 2 billion years ago. There's a period of time in terrestrial evolution called the Great Oxidation Event. I'll wait till that passes. So there was no animal life or plant life 2 billion years ago. What there was microbial life. And the oceans were probably green with cyanobacteria that had learned how to do photosynthesis and emit oxygen as a byproduct. So that's called the Great Oxidation Event, and if it hadn't happened, we would never have gotten past bacteria in the evolution of life on Earth. So there's a volcanic heat as an energy source. There is a salty ocean. There's probably a global ocean, more or less like it is now, except we have continents now. Back then, there was probably no land masses except volcanic land masses coming up out of the ocean. And finally, and most important, there's another kind of water than salty water. Salty water was being distilled into fresh water, and the fresh water forms pools on volcanic land masses. Now, I'm a chemist, I'm a biochemist. I would never go to the ocean to get salt water to do an experiment with if it involves biochemistry. The reason is that the salts in seawater are toxic, particularly calcium's toxic, because it can bind to phosphates and produce an aggregate. But in the laboratory, we use distilled water. So keep in mind that there is a source of distilled water on the early Earth as a result of rainfall. That's very important because that forms those puddles I talked about. Well, what we want to do now is to go through 4 statements. For life to begin, organic compounds must be able to self-assemble under natural conditions. Now, I'm a little different than many of my research colleagues because I like to step out of the laboratory and see if what I do in the laboratory actually works out in the wild. So I visited volcanoes all over the Earth: Iceland, Hawaii, Kamchatka, New Zealand, and even Bumpus Hill in California. That's on Mount Lassen. So these are sites that are available to me as a research chemist, and I'm going to do an experiment on Kamchatka that tests Darwin's idea. So we visited Kamchatka along with these other places. The geothermal regions in Russia, in eastern Russia and the Kamchatka, these are plausible models of the primitive Earth. So when I was in a volcano looking down, I reminded myself, this is what the Earth was like 4 billion years ago. Now, as a chemist, what can I do knowing that and knowing what the Earth was really like? So I had a sterile place because of the high altitude. We were working about 6,000 feet. High latitude, we were up quite high toward the Arctic area. And recent volcanism actually sterilizes these sites. There's nothing alive there except us and a few microbial populations. So what I'm going to do now is to show you how to get to Petropavlovsk. Petropavlovsk is a city of 160,000 people, and it's basically the city on all of Kamchatka. And if you look at Kamchatka, you see it's a peninsula, and it's about the area of California. You can imagine California settling down on that. Just right below Kamchatka, that's where Japan begins. So the Japanese islands are extension of that peninsula coming down. So to get to Petropavlovsk, you hop on a plane in San Francisco, fly to Anchorage, Alaska, and then next morning you get on another plane, a Russian plane, and 5 hours later you're landing in Petropavlovsk. Now Petropavlovsk is a weird sounding word, but if you know Russian, you know that that's Peter and Paul. Vitus Bering explored this whole area and discovered that bay, and his 2 ships were St. Peter and St. Paul. So he named that bay Petropavlovsk. That's what it looks like from the air. This is an artist's rendition of a satellite view. Kamchatka is the most volcanic area on Earth. It has a string of 20 or 30 volcanoes going right up the center of the country. You notice Petropavlovsk with that little yellow center there, a little yellow word, and then just below it you'll see Mount Mutnovsky. Mutnovsky translated is Mount Smoky. So it's interesting. This mountain is always smoking because there's glaciers melting down onto this hot magma that boils the water. So we have that constant roar of boiling water in the background when we're working there. So here is Petropavlovsk. It's all new. Basically everything you see was built in the last 50 years. There are no ancient cathedrals or churches because there's nobody there. This started out as a Russian military base. It was a submarine center for Russian submarines during the Soviet era. But it's now grown into what amounts to a place where tourists come through to see Kamchatka. I highly recommend Kamchatka. It'd be well worth a vacation if you can get to it after the tension between us and Russia settles down. That volcano is right near Petropavlovsk. It's called the Koryatsky. You can see it on the horizon. And now this is looking down a Google map of Mount Mutnovsky. So we would take a 4-wheel drive vehicle, we as being myself and 5 other scientists that was on this particular field trip. This was in 2004, 20 years ago. And this would go overland on little gravel roads and finally actually on the volcano itself. And we were deposited about 2,000 feet below the crater. There's a little wooden shack there. And for a week or 2, that was our home, and we would go up into the volcano. So this is what it looks like to be in an active volcano. And it had erupted, had a minor eruption a few years before, and I was there, and that's me sampling a boiling mud pit called a fumarole. Fumes are coming up, that makes it a fumarole, and those fumes are a mixture of water vapor, of course, but also hydrochloric acid, hydrofluoric acid, and sulfur dioxide. That's that sulfur smell that you smell everywhere when you go visit a volcano. So I took that mud sample home and studied it, and there's nothing there that is organic. There's no amino acids in parts per billion, nothing alive, of course. So that is not Darwin's hot little mud puddle. We looked around on the volcano and we found something much better. And I call that Darwin's hot little puddle, and that's the experiment we're doing to test that idea of a warm little pond. I'm pouring into it a mixture of 4 amino acids, 4 nucleobases. These are the ones of RNA, adenine, uracil, guanine, and cytosine. An amphiphilic molecule, you know what that means now. It's a fatty acid called myristic acid, some glycerol, and some phosphate. So that was my example of what I thought maybe a prebiotic soup would have in it. It's white because the lipid, the fatty acid, forms an emulsion, so it gives it that milky look to it. And I simply dipped the water out of that hot boiling spring and mixed it with the dry material to make that solution. We found some interesting things, and if you're interested, 2 years later, we published our findings in the Philosophical Transactions of the Royal Society. And that, by the way, is another hydrothermal area on Mount Lassen in California, Northern California. And maybe some of you have visited there. It's really quite spectacular place. What we found would have disappointed Darwin. All of the stuff that we added stuck to the clay that is circulating in that puddle. You can see there's a boiling center, and it's just keeping the clay circulating. It literally disappeared as reactants, and that was kind of disappointing to us too. I'd hoped that maybe some of it might polymerize, for instance. But what I saw that was real happened in just a few minutes, and that is a white foam appeared all around the edge of this boiling pool. And I knew from other work that I had done that that foam was membranes. That fatty acid that I had there self-assembled into membranous compartments, and this is the one thing that actually happened there that we took home as a lesson to be learned, because this pool undergoes wet-dry cycling. If you look at that bigger rock down there in the lower right-hand corner, you see at the top is dry, and then there's a sort of a reddish, moist area. And all those other rocks around there have that dry top and a moist, reddish area. So this is wet-dry cycling in action in this pool. And that stuff that I dumped in is going to go through a wet-dry cycle as that pool further evaporates. So I am going to try to convince you that this is a plausible place for life to begin, and that Darwin was on the right track when he speculated about a warm little pond, but there's more to the story, as you will see. I went home inspired by what I'd seen, and I built my very own wet-dry cycler. This is a plastic container filled with carbon dioxide. You can see the tank there. And there's also water coming in. There's a little red perfusion pump down the lower left. And that's dripping water into two little wells that you can see at the top and the bottom of that disc. That disc is rotating, and each of those wells goes through a wet and then a dry cycle every hour or so. So if we set that up with little glass vials holding the reactants, we come back next morning and our experiment is done. There's probably been about 8 cycles total there that we can analyze. There's 2 heat sources. They're just ordinary laboratory heaters with at 80 degrees C. Well, there's one thing that I've got to teach you. If you haven't had a course in chemistry, you have to know this in order to understand what I'm going to tell you next. Ester bonds are synthesized by removing the elements of water from between an organic acid and an alcohol. So this is one of the first things that I did as a chemistry major do. I made ethyl acetate by mixing ethanol and acetic acid. A spontaneous reaction occurs, and the result is ethyl acetate and water as a product. That's called a condensation reaction. And that little red atom there, labeled ester bond, that is in fact an ester bond. What nobody ever thought of, as far as I can tell, is that you can do the same thing with a monomer of a nucleic acid. So that monomer is the A of DNA. There's 4 monomers: adenine, guanine, cytosine, and thymine. And those 4 carry the code of life, and we are here. Everybody sitting in this room, including me up here, we were designed around the genetic information in our DNA when we were a fertilized egg, half from our father, half from our mother. And that is, of course, discovered back in 1953 by Watson and Crick, and they realized that DNA is a double helix. And that began the revolution that we're still living in, and that many of you in the room are in fact using in your jobs, is the fact that we can now control so much of the life process because of the fact that we understand DNA and RNA in a way that was never before possible. Well, what's the last thing I want to show you here is that if you want to pull the reaction to the right and get more ethyl acetate, you just let the water evaporate. If it goes away, that moves off to the right and you get more ethyl acetate. So what I'm doing when a little puddle dries out is I'm allowing water to disappear, and I'm pulling the reaction from left to right and making long strands of nucleic acid. And it's as simple as that. I really do think that nucleic acids came to be, emerged on the early Earth before life, and that life began when nucleic acids were encapsulated in a lipid membrane. And that's going to be the last part of my talk. So we tested the hypothesis by making up a solution, 10 millimolar, if you know concentration, 10 millimolar mononucleotides. We made them acidic. pH 3 is about the acidity of lemon juice. It has to be acidic because this is an acid-catalyzed reaction, the condensation reaction. We got products after multiple wet-dry cycles, and we did nanopore sequencing. I was so pleased when I walked into the exhibits, and there was Oxford Nanopore Technology. I've been involved with that company since its start, and we have worked together. I was on the advisory board for quite a few years, and I'm now using one of the products of Oxford Nanopores, and it's really great to see that company here at this conference, and a big surprise. Okay, let's see what we learned from using nanopore sequencing. A little bit of history here. The very first nanopore, and we invented that word, by the way, to describe this particular nanopore. It was called hemolysin. This is a toxic protein put out by Staphylococcus. And it's called hemolysin because it hemolyzes red cells. If you add this, not very much, doesn't take very much, just a few molecules per red cell, it punches a hole in the membrane and the red cell swells up and pops and lets all its hemoglobin disappear. That's called hemolysis, and hemolysin got its name from that. If you look over to the right there, you can see a strand, a single strand of DNA or RNA, and it's an almost perfect fit to go through this pore from the top to bottom. I'm going to show you a little video here. This is a molecular dynamics that's been around now for quite a few years. It was done by Alexei Aksamentiev when we first began to work with this, and he loves to do these kinds of videos. So we now know that if a single-stranded nucleic acid gets started into a pore, and if we have a voltage across that pore of about 1/10 of a volt, between 1/10 and 2/10 of a volt, something amazing happens. And this is what happens. The voltage pulls the strand through by electrophoresis. This is single molecule electrophoresis. And if you had a sensor built into that protein that would capture the ionic current that is going through the pore along with the DNA, you would discover that in fact the DNA blocks the ionic current to about 90%, okay? And that's basically what these devices do. There's a MinION. That's the first one that came out in 2014. I brought one. You can come up and take a look at it if you want. Not much to it. This cost me $1,000. I carried it around in my pocket. It's been up on the International Space Station. It's been down to the Atacama Desert and every place. This went to Africa during the Ebola epidemic, and the workers, the scientists there, were able to follow the course of the endemic as it moved from one little town to another, using something just like that. We are now using the Promethean, and the Promethean, let me show you something, has the Guinness World Record of doing an entire human genome in 5 hours and 2 minutes. This is literally in the Guinness Book of World Records as the fastest human genome ever sequenced. It was done at Stanford University. It was done on patients that had genetic diseases, and the doctors were trying to find the mutation that was causing the disease. And they found it using the Promethean device. So that was published about 3 years ago. And now I want to show you the molecular structure that is doing the work in the Promethean and other MinION devices. Here's a double strand of DNA. That's just something that you would isolate from any living organism. This is a helicase enzyme that unzips the DNA double helix and allows one strand to go down through the pore. And there's a voltage of about maybe 180 millivolts pulling this downward. The helicase is a molecular motor that ratchets the DNA into the pore. It's not a kind of a smooth motion, it's this kind of a motion. And you can see it off to the right there. Each of those is one base moving into the pore near the sensing center. And I think that's probably an actual recording from one of the works. The company also has a proprietary CSGG nanopore that they are using in their instruments. So the company, by the way, did some spectacular things. In the laboratory where I was working, we had one nanopore in a very fragile lipid bilayer membrane. It might last for an hour or so. The company said, we're going to need to send this all over the world by shipments. How can we get something tough enough to stand up to that? Because a lipid bilayer would not. So they invented a membrane that would stand up to the rigors of shipment. They also invented a new kind of a pore. This is the third pore after hemolysin. There's one called MSPA that Jens Gundlach discovered up at University of Washington. But this turned out to be the best one for our purposes. And that's the little area there that actually senses the ionic current flowing through the pore. And notice that current is in picoamps. You know, nanoamps are a billionth of an amp, but a picoamp is at the trillionth of an amp range. And that's the current of 700 million ions per second passing through that pore. Chloride, this is in 1 molar potassium chloride. Chloride goes this way, the potassium goes that way, and that's the ionic current that's being measured. 700 million ions per second for the hemolysin pore. It'd be a little bit different for the CSGG pore, but certainly that is just a number to keep in mind. So that's what's happening when we sequence DNA in a nanopore. Here we see the DNA solution being put in. It's slightly yellow because one of the ways they turn the current, which is an ionic current, into an electrical current, is that they have ferricyanide as part of the solution. And that has a redox reaction that turns an ionic current into electrons that are then measured by the laptop software. Okay, this is a Promethean that looked at the nucleic acids that we were making by wet-dry cycles. I don't think anybody would have believed that this would be possible. Me either. I was really totally taken aback when we discovered that nanopore sequencing can measure the sequence of stuff that never saw anything alive. It's just mononucleotides self-assembling and then zipping up by ester bond synthesis. So if we put in TMP, that's thymidine monophosphate, we get a whole bunch of T's as homopolymers. This, by the way, is a little marker sequence that we use to make sure that this is a real sequence. If we have a mixture of AMP and TMP, we see this sort of thing. We see homopolymers of A, and sometimes they're attached to a homopolymer of T, but we don't see a random mixture. We don't know why that is. We're sort of puzzled by that. We expected to see a random mix, but we don't. So it tells us something about the process of polymerization, I think. If we put all four in, finally we see what we expected, and that is a mixture, a random mixture of bases making up a sequence. These sequences are about in the 100 nucleotide range, and they can be much longer. We can see them by atomic force microscopy, and there we're seeing long strands, thousands of nucleotides in length. So you can make as much nucleic acid as you want just by putting mononucleotides into a wet-dry cycling process. And if you want to start a company using that, we have 2 patents available for licensing, so see me after my talk. Okay, well, I think I mentioned something that is we need to have a cellular compartment. That's easy. All you do is to mix in an amphiphilic molecule, like a fatty acid, for example, and here I'm mixing in vesicles of fatty acid, and the little small circles are any polymer that you want. It could be DNA, RNA, or protein, or small molecules. When you put it through a single wet-dry cycle, the lipids fuse into a multilamellar matrix, it's called. So those are all lipid bilayers, and look, the polymer that we had to capture is now trapped between those lipid bilayers, and we have X-ray diffraction analysis of that, and that's real. That's the way it is. Then when you make it wet again, look, the polymers are now inside the vesicles. It's really pretty amazing. It's that simple. Any of you could do this in an afternoon if you had a little DNA and a fatty acid or a phospholipid. So it's the easiest thing. I'm pretty sure this is probably how the first cells came to be. So I'm going to show you the evidence that that's real. Up here, we have a mixture of a fatty acid, decanoic acid, that's 10-carbon fatty acid, mixed with its monoglyceride. And if you do a 1-to-1 mix, you get beautiful membranes forming. You can see them over here. We have AMP and TMP present. If we put it through one cycle, we begin to see some fluorescence inside these membranes. And the fluorescence is DAPI. That's a fluorescent dye specialized for DNA. Here's 2 cycles, 3 cycles, and 4 cycles. These cycles are maybe half an hour wet dry. And look, we get these things that we call protocells. The DNA has aggregated inside the membrane, and it's on the way to becoming a population of protocells that can undergo selection and evolution. So that's the answer to Charles. I would say to him, we know how to make protocells by the trillions, and your idea of selection and evolution would work on those, because some of the protocells are going to be more robust for one reason or another. Some of them might even have a ribozyme activity that can make more of itself. That has been done by another colleague. So here's how we think life began. And you're hearing this for the first time because this has not really gotten out there into textbooks yet. We think that wet-dry cycles are very important. We think that life did not invent nucleic acids. Instead, the nucleic acids were being synthesized ahead of the origin of life. If the amphiphils are also present, they capture the polymers inside vesicles, and you have protocell populations being produced, and each cell is different from all the rest, and that's very important. Darwin said, populations with variations. That has to give something that can undergo selection and then be amplified by growth. So the proposed alternative that we are now testing is that life began when selected systems of encapsulated polymers emerged that were capable of catalyzed metabolism, growth, and replication. So we're on track to make that happen. Last slides. Mars. I want to send a nanopore to Mars. I'd love to get some of this frozen water that we can see by satellites circling Mars. You could go ice skating on that. That's the water left over from when there were oceans of Mars. You can even see on the edge of craters layers of water that's a mile deep. The subsurface has a lot of water there frozen. I would want to have NASA figure out how to drill down to that water. I would like to bring up a sample of the ice, melt it, and put it into a mid-ion and see what I see, okay? I would love to see nanopores discover life on Mars. Okay, this is a summary slide. This is your take home. You now know that organic compounds are synthesized on interstellar dust particles from which solar systems emerge. You know that they can be delivered by infall. 30,000 tons per year is still being delivered by infall of these dust particles. They accumulate on volcanic land masses, on Mars, on Earth. They go through hydrothermal cycling that I've talked to you about, and the result is protocells that can now take the next step toward life, selection and evolution, toward a true living system. Okay, that's what I have to tell you today, and I'm happy to take any questions.
So if you are going to ask a question, I ask you to come to one of the two mics so that everyone can hear the question and the answer. Why am I not surprised?
Here comes a volunteer.
We got two. Pick up the pace, come on.
Thank you for the excellent talk. I was wondering if you could comment on the RNA world.
Speak a little more slowly, please, so I can get it.
I was wondering if you could comment on the RNA world theory and what roles you think RNAs had in either catalyzing the creation or use of DNA and proteins.
Right. The RNA world was a phrase brought up when ribozymes were first discovered. Tom Cech and Sidney Altman discovered that RNA can have catalytic activity. It's a cleavage activity that RNA, certain kinds of RNA can do, so it's self-cleaving. So they discovered ribozymes, and Walter Gilbert at Harvard immediately said, maybe there was an RNA world before the world that we now live in, RNA, DNA, and protein world. So that has stuck, and it's a good working hypothesis because it suggests experiments. One example is that our ribosomes, when we dig down to the center of the ribosome where proteins are synthesized, that's a ribozyme, maybe left over from a time when ribozymes were involved in the main biomolecule involved in life. So that is where the RNA world came from. It has multiple tests that are happening. The ribozyme, the one that I'm most interested in, was discovered by Phil Holliger, who's a British biochemist. And he's evolved a ribozyme that can copy itself. This, I think, is very important. And it's a step toward, a major step toward what we call life. Okay?
Thank you.
Yep.
It's a fantastic talk. I appreciate that.
Thank you.
Congratulations. It's well-deserved. I'm wondering about analog or alternative backbones of DNA. We spent a lot of time theorizing about it, and it's all phosphate, but in all of your years of thinking about this, are there any possible alternatives that could serve? That's
Yes.
the first question. Sorry, I'm going to make two from one. And then the idea of having sulfonated compounds struck within the DNA.
Yeah, good question. Steve Benner has been playing around with alternative bases to see if he can get alternative bases as part of a nucleic acid structure that actually assembles and works. And he's found a few that can be introduced into a nucleic acid and kind of be functional. Albert Eschenmoser, though, maybe 20 years ago, began to experiment with all kinds of different ways to put alternatives to DNA together. And as you might imagine, one of his findings is that the DNA we now use after several billion years of evolution is about as good as it gets. None of his others had the kind of functionality that our DNA does. So I think most people would say, yes, life played around with other ways to make nucleic acids. But out of that was selected 4 bases that actually we use now, both for DNA and RNA. RNA has 4 bases to uracil replacing thymine in RNA. Does that get at the sense of your question?
Yeah, I was actually thinking about the backbone, not the bases. And I've seen some theories like fluorine could possibly be an alternative. It's not exactly my field, but it's worth pondering. And then the other thing I wanted to get your thoughts on is sulfonated compounds linked to DNA.
Yeah.
The ancient world is highly sulfonated, and it
It
seems
sure was.
odd that it's not somehow involved, whereas we have methylated DNA, but not necessarily, there's no sulfur links that I can see.
Mm-hmm. Yeah, you may remember a young woman who thought that she had arsenate as a replacement for phosphate in metabolism. And she was convinced she had some of the evidence, but not all of the evidence. And that idea came and went in about 6 months' time. And she's still working. She has a scientific career. But that's one example of a replacement for something in the backbone, is maybe why not arsenate? The problem with arsenate is that it hydrolyzes so quickly that it could not possibly be part of a permanent molecule. That turned out to be the problem with it. Okay, so what else? Sulfate. And why isn't sulfate more widely spread in biology? You know, just sort of an add-on to things like cysteine with its sulfhydryl group, you know, and various sulfated proteins, you know, as part of that. But it just doesn't have the chemical properties that phosphate has, for example. Phosphate has this wonderful property of being relatively easily attached to another molecule by an ester bond. And sulfate doesn't have that property. And also, we have to turn over our polymers. So there's got to be something that can hydrolyze. And phosphate is the ester bond that can be hydrolyzed by a phosphatase enzyme, for example. So a lot of what we see now in life has had billions of years of evolution to get it right. And here we are, you know, conscious organisms. pretty amazing.
Sometimes.
Yeah, oh yeah, right.
Depends on the beer that night. So you've done enough puddles to get trillions and trillions of combinations of RNA.
Yeah.
You've encapsulated it in a membrane, but now you have to get out of the puddle. You have to persist, right, in order to replicate. You could fill up that one puddle with something that's self-replicating, but that's still not going to lead anywhere if the whole puddle gets evaporated or dries or goes away. So are these membranes tough enough to survive being flushed into salt water, or are they, what's the next step?
Oh boy, my favorite question. The calcium of seawater is toxic to so many biochemical reactions. Our cells pump calcium down to micromolar concentrations. And when I want to do this, I release calcium and it activates the contractional process. But that goes from micromolar to maybe 10 or 100 micromolar, something like that. And then it's immediately taken back in by the sarcoplasmic reticulum. So calcium is a real problem with seawater. The reason is that it cross-links carboxyl groups and phosphate groups into insoluble aggregates. And I just see this, when I've tried to work with salt water, I just saw that happening, and I just couldn't get around it. So I think the ocean, I think that the first life probably began in freshwater, and it's all downhill to the ocean from there. So you would have a chance to go through multiple evolutionary steps to the point when it could finally withstand ocean water as an extreme environment for the early life.
So is this still happening? There are volcanoes, there's influx of dust.
So
I guess there's a
is
lot
this-
more oxygen around, but is
Yeah.
this still happening right now?
Yeah, I think so. I'm sure that there are organisms evolving to survive the jump. Salmon, of course, easily have evolved a mechanism to get from salt water to fresh water.
I'm thinking more earlier. Are there still
even earlier,
new
yeah.
RNAs being synthesized? Well, they're not new anymore, but at least new polymers being formed in puddles on volcanoes right now.
Yeah, okay, I agree. Thank you for the question.
One last question.
Alrighty, hello, thank you. Fantastic talk. You know, there is evidence of amino acids being present in space in things like comets and asteroids and whatnot. And I want to hear your thoughts on how that kind of, the panspermia hypothesis or model of life with amino acids falling to Earth, how that might fit in with kind of the context of this nucleic acid-based model.
Okay, condense that into a simple statement because I didn't quite follow all of that. I'm sorry.
Sure. So an alternative model for life on Earth, right, is panspermia from amino acids that are present in space, comets, asteroids, things like that. I wanted to hear your thoughts on that kind of amino acid-based model of beginning of life versus what you talked about today with the nucleic acid.
Okay, you're proposing that we incorporate amino acids at some point into this story that we're telling. Is that the main point? I agree. A ribosome is probably the core of life as we know it. And a ribosome had to have a primitive ribosome. And Lauren Williams at Georgia Tech has a nice evolutionary story starting with a short peptide and a short oligonucleotide. And those happen to have if they were together, they happen to have the ability to do the kind of reaction that we now associate with protein synthesis.
Alrighty, thank you.
Yeah.
Okay, at this time, I'd like to present you with the award.
Hey, let's get going. Oh, okay. Oh, yeah. Oh, like that? Good. Okay.
Are we good? All
Super.
All right, let's thank Dave one last time.
Beautiful.